Okay, so our next speaker is David Mark. He is a third-year PhD student in Dr. Paul Herron's group at the University of Strathclyde. His research focuses on characterizing the anti-pseudomonad activity of Micromonospora isolated from hyper-arid environments of the Atacama Desert.
Hi, hi everyone. Yeah, I'm David. I'm a third-year PhD student at the University of Strathclyde, as Poppy said, and thanks very much for having me. I focus on finding out why these hyper-arid Micromonospora are able to kill Pseudomonas aeruginosa, and I thought I'd start off by talking about Pseudomonas aeruginosa and why it's such an important pathogen for the discovery of new drugs. And apologies for showing these images just before lunch, but I think it's important to remember how horrifically infections that these pathogens can cause. It's a gram-negative member of the gamma proteobacteria and a major opportunistic pathogen which targets cystic fibrosis patients, immunocompromised patients, and patients with compromised epithelial barriers such as burn victims. And part of the reason it's so hard to kill is because it's intrinsically drug resistant, and it's really just phenomenal at keeping drugs it doesn't want in the cell out of the cell. It has a phenomenal export system, and it's also capable of losing the proteins that would otherwise allow drugs into the cell. Traditionally, natural product drug discovery has been digging up your back garden, seeing what actinobacteria, particularly streptomyces, you can find and screen them for activity. You've got a wee zone of inhibition there. And part of the problem with this approach has been rediscovery in the face of increasing antimicrobial resistance, which is why we turn to less well-studied organisms. And my personal favourite is Micromonospora, which I refer to as the least rare actinomycete. It is the second most commonly isolated actinomycete after streptomyces, and we already know it can produce clinically relevant drugs such as gentamicin, but also really potent anti-eukaryotic molecules such as calicheamycin. It has a complex life cycle which is slightly different to streptomyces. In grey here, the biggest differentiator is they don't actually erect aerial hyphae that differentiate into spore chains. They just form monospores at hyphal branch points. And I'm going to talk about 3 Micromonospora which were isolated from the hyper-arid environment of the Atacama Desert. Micromonospora species 03, 05, and pH63. When we screened these guys against Pseudomonas aeruginosa, we found that 03 and 05 that produced zones of inhibition against pathogen, which ideal. Moving forward, we sequenced the organisms and discovered that we have 2 new species of Micromonospora, which is great. Interestingly, 05 and pH63 on the left by ANI are the same species, which raises the question, why is one exerting anti-pseudomonad activity when the other isn't? Whereas Micromonospora species 03 on the right there is its own novel species. And when we put the genome assemblies through anti-smash, you see a real pick and mix of biosynthetic gene clusters. I imagine you've had your fill of chord diagrams for today, but I just want to go through how diverse the BGCs present in these organisms are, and it raises the problem of how do you prioritize which clusters to investigate. I'm going to focus now on the best assembled genome we have, which is that of Micromonospora species 05. And it's a really beautiful organism. This plate is a week old, and a week later you can see the characteristic black spores developing. Using a combination of Illumina and nanopore reads, I've managed to get a single circular scaffold of the genome, and you can see it's BGC rich with 23 clusters scattered across the genome. when you have 23 clusters, the question is then, which is the cluster? And as a bit of a lockdown curiosity, I thought, can we compare the biosynthetic gene cluster landscape in other Micromonospora to help prioritize which clusters are important? To give you a quick overview of actinomycete genetics, streptomyces have linear genomes, and the biosynthetic gene clusters illustrated here tend to cluster towards the arms of this linear chromosome, the arms being approximate equivalents of the termination point, the replication terminus in circular chromosomes of bacteria. So we thought, if BGCs cluster towards the arms in streptomyces, do they cluster towards the terminus in Micromonospora? So I pulled 9 complete Micromonospora genomes from NCBI. I wanted to use complete genomes because I wanted the assembly to be as reliable as possible. To that end, I actually excluded Micromonospora species L5 from this because there was quite a large inversion, which made me suspicious of it. To compare the layout of biosynthetic gene clusters, took the circular chromosome assemblies, linearized them so that REC was at either end of a plot with the terminus towards the middle. And when you plot biosynthetic gene clusters along this line, you can see 3 humps on the left and right next to REC, and then a real very diverse array of biosynthetic gene clusters towards the middle. And I found it interesting that the more conserved biosynthetic gene clusters were closer to the origin of replication. So I want to talk to you about these 2 terpene clusters, this NAGGN cluster, which is an osmolyte, and this type 3 polyketide synthase, all close to the origin of replication. When you look at the actual architecture of these gene clusters, you can see that they are really highly conserved. I don't want to get into the actual gene content of the clusters too much, as much as if you look at the identity bands, you can see that they are identical clusters near enough, and they are always found at the right of REC. It's the same for the osmolyte cluster, and it's not just on the right-hand side of the cluster. You can also see it with the type 3 polyketide synthase, and this is part of the inversion I was talking about with Micromonospora species L5. But why is this important? It's worth remembering that biosynthetic gene clusters haven't evolved to create drugs for us. They've evolved to give selective advantage to the organisms. I'd argue that clusters being close to the origin of replication means that they've been selected to be vertically transferred, whereas towards the terminus maybe implies a role for horizontal gene transfer. I'm not honestly sure why you see this bias, but I think it's also important to remember that high quality genome assemblies can help prioritize clusters of interest when you start to see where they fall on the genome. And I really want the takeaway from this talk to be is where the BGC is matters as much as what it is. In the future, I'm going to start investigating what these clusters are by cycling between mass spec to get molecular leads to link the clusters, disrupt the clusters, and see if we lose the mass spec peaks. That'll be the next year of work for me, and hopefully it goes well. And finally, I'd like to thank the microgroup. This is a Christmas party last year, pre-lockdown, and just a wee memory of the good times. I'd like to thank my supervisor, Paul Herron, our collaborator in Antofagasta, Dr. Christina Dorador, and my second supervisor, Nick Tucker, as well as their respective groups for being phenomenal support, as well as the group of party animals that is the SIBS microgroup you can see there. Thanks very much for listening.
Thank you, David. It was a really great talk.